to open this. Okay. So, let's see. Where were we? Lymphocyte development. I know we talked about affinity maturation and class switching. I think we probably... Oh, yeah, we even watched this video, right? So how is it that we can make all the different antibodies, and even the T-cell receptors are made this way? We literally shuffle up our DNA, right? We pick basically our lottery numbers, right? They pick different sections of the chromosome, right? And so once they do that, they're programmed that way. That's all they can do. So that's the gene rearrangement, right? So they're literally rearranging and picking their genes. The imprecise joining they don't show in this animation, and that is still something I'm struggling with trying to explain to you guys. And I actually think I've come up with something, so I'm going to try and draw it out next time for you guys. I don't want to spend time this time, but I think I've got an example that'll make sense. So I'm excited about that. And then combinational, because remember, antibodies and even T-cell receptors are made up of two proteins. For antibodies, it's two copies of two proteins, right? So it's those two copies that come together, the light and heavy chains in the variable region, that create that variability for each antibody or each T-cell receptors for T-cells. So that's what they mean by combinational variation. So those levels of variation, and then they're going to do little tweaks, right? As they're become activated, they're gonna cause mutations in the variable region in hopes of becoming more, have better affinity, better binding to the antigen. So for both B cells and T cells, they recognize a distinct, right? Because each B cell is different that comes out of the bone marrow. Each T cell that comes out of the bone marrow is different. But they need to be checked out to ensure that they function properly. The B cells undergo this in the bone marrow, right? But T cells undergo this process in the thymus. And at one time I did have an animation for that. I'm going to see if I still do in Canvas, because that one I think is worth playing. So let's look under my animation list. So module 2, animations and websites. Right here, Tcell Development. So let's see. Let me plug this in.
Mature and differentiate. T-cell development takes place within the thymus, a specialized organ where T-cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations, and as thymocytes mature, they pass from one environment to another. T-cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start their process of maturation by rearrangement of their T-cell receptor genes. As maturation proceeds and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T-cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocytes express both CD4 and CD8, as well as an alpha-beta T-cell receptor. A cell that has a receptor...
So, MHC class 1 and class 2. I mean, CD4 and CD8, what do those mean to us? Right, our helper T cells, right? CD4 are helper T cells, remember 4H, right? 4 is helper, right? So CD8 cells are what type of T cells? Cytotoxic T cells. So when they're born, as I said, they have both, but it's going to be determined which one they're going to get the signal to continue making, right? So there's going to be this choice. Are you going to be a helper? Or are you going to be CD8, cytotoxic. Make sense? Okay, so that's a very important point, right? Knowing those markers and which T cells have them. Can't stress it enough. Guarantee you it's on the test.
Are able to recognize an MHC class 1 molecule receives both a survival signal and a maturation signal.
So yeah, I forgot. So the next step, how do we determine which one it is you're going to keep? It depends on whether it can recognize the molecule it's supposed to recognize. So remember, on our cells, we express MHC class 2 and class 1. Which one goes with which, right? So CD4 matches up with MHC class 2. 2, 4, 6, 8. Who do we appreciate? Right? That's how I remember that one. I sing that song. Okay? So MHC class 2 and CD4. All right, and remember again, helper T cells help what? Cells. But a special set of cells. B cells. B cells is one of them. And the reason why is B cells do something called antigen presentation, right? They take in the antigen and they present it. So that the helper T cells sometimes will help them make that transition into the plasma cell and memory cells, right? And activate it so it proliferates. So an antigen presenting cell. What's another one that takes in antigen from the outside and when they would present it to a helper T cell? The dendritic and the macrophages, right? These are the only guys, these are specialized antigen presenting cells. These are the only ones that have MHC class 2. They're the only ones that are going to communicate with helper T cells. So the other one that we have, of course, is CD8, and it talks to MHC class 1 then, right? Because we've already talked about 2. That's found on all your cells, right? All your cells, because that's your defense against stuff getting in or stuff going badly on the inside to be able to express that information outward. So it makes sense that we would communicate this to cytotoxic T cells, right? If something gets inside of our cell or if our cell is not doing what it's supposed to do, what do we want to do to that cell? We're going to let it be? No, we want it killed, right? That's why we want a cytotoxic T cell, right? To recognize any one of our cells that are infected or behaving badly. We don't need help in this situation, we need death. Does that make sense to you guys? Okay. So we'll go through, and that's what they're going to do in this process, right, is make sure that these matches happen and happen correctly.
Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class 2 molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class 1 or MHC class 2 molecules fail to receive any survival signals and die by programmed cell death or apoptosis. Thymocytes that are able to recognize MHC class 1 or MHC class 2 peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature, naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system.
Where are they going? Where are those T cells going to go? What's home for them? Spleen is one of the homes, right? That's one of our secondary lymphoid organs, right? That's their home. Now, if we're talking about the lymphatic system, it's littered with a whole bunch of homes for T cells and B cells and macrophages and dendritic cells. What do we call those? Lymph nodes, right? What about up here? What extra protection do we have in our throat? What do we have? Tonsils. Tonsils, right? You have your palatine tonsils and your lingual tonsil, and then you have the one behind your nose, too, in the top of your throat, your adenoids or your nasopharyngeal tonsil, right? And then you have skin, associated lymphoid tissue, right? So all along your skin, you have clusters of B cells and T cells and macrophages that can respond right on site, lining your whole mucosal layer of your intestines, right? You have mucus-associated lymphoid tissues. So they're all over the place, but there are large concentrations in those lymphoid organs, secondary lymphoid organs, in your tonsils, in your appendix as well, right? In these areas. So they, though, have to go through this selection process, though, in the thymus first, right? We'll just close that out. So this one will be a good review. I will actually play this one as well. Not to bore you guys with too many videos this morning, but I find sometimes they help to see the visualization of going through the steps. So this would be somewhat review. So think about this too, right? This is the steps in the process. And this is a very important process that we're going to keep talking about, right? So I think one of the things in the Learn Smart that I did was kind of putting stuff in order, right? And that's important, understanding the steps, right? When we talk about how does this process work, right? Understanding the steps in the process. Because when we get to disorders, right, it's going to be the same steps. The problem is the outcome is not what we want, right? For that particular substance that we're dealing with.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T-cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T-cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated.
Okay, so that's the T-cell receptor. What's this molecule right here? This is a helper T-cell, right? That's the CD4. Because as you guys saw in that other video, that actually interacts with the MHC class 2. This is how they know, okay, these are the two that match up, right? These are the two receptors that can communicate with each other.
The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells.
Does a B cell always need that helper T cell to produce antibodies? So notice that this said it was a T-dependent antigen. So in this case, for this antigen, the answer is yes. But is it always yes? Now we have what's called T-independent antigens, which means that the B cell doesn't need help from the T cell. It can do it on its own. But it's limited, right? It'll only produce the first class of antibodies that we make. What's the first class of antibodies we make? IgM, right? And how is that one, what does it look like when it's secreted? Yeah, it's a big, huge pentameter, right? Remember, mega big. I always remember M for mega big. So that guy's limited, right? Pretty much stays in the bloodstream. He's only going to get out into the tissues really with inflammation or something like that. Doesn't cross the placenta. Can activate complement, right, which is found in our blood. But really, that's about it, other than cross-linking, right? No, that's not true. Can neutralize toxins and things as well. We can't do all the antibody antigen outcomes. So T independent, right? So this is when B cells can be activated without T cell help or T cell help. And this what has to occur for this is the B cell receptors, which are basically its antibody, right, of IgM class on its surface. A lot of them have to interact with the antigen. So not just a couple, but a whole bunch. And this is commonly referred to sometimes as string response. In highly repetitive molecules that have pieces of them that are the same, right, that repeat, have a tendency to be able to induce B cells without T cell help. So an example are polysaccharides. So poly meaning many, right? Saccharide refers to sugars, right? So lots of bacteria have sugars associated with them, especially lipopolysaccharide, right? Which we know is a component of gram-negative cells. These are all examples of T-independent antigens. And again, because they're able to interact with a lot of the B cell receptor, the antibodies on the surface of the B cell, that's enough of a signal to get it to activate. But again, it's going to stay at IgM. It's not going to switch to the other classes. That happens when you have helper T cell involvement. But sometimes, hey, that's enough, right? So what are the markers found on all nucleated cells and antigen-presenting cells? We call these, it's an abbreviation, it starts with an M. Yeah, MHC1 is on all nucleated cells, right? And what's found on antigen-presenting cells? MHC2. What's the important relationship between T cell receptors and CD markers? It tells you basically what the T cell is, right? So CD4 is what? Helper T cell. CD8, cytotoxic. We already watched antigen presentation animation, I thought, maybe? I don't know. So, dendritic cells. So in the animation, we saw what cell activated the T cell? Macrophage. But most of the time, it probably is dendritic cells, right? Dendritic cells are the major ones in T cell activation. Same process though, right? It's going to take stuff in, it's going to digest it, it's going to present it on MHC class 1 or class 2, depending on what it is, right? What that antigen is. Do we need a cytotoxic T cell response? Then it's going to be MHC class 1, right? If we need a helper T cell response where we're going to maybe activate B cells, then we need a CD4 helper T cell response, which means we have to present in an MHC class 2. Right? So stuff that gets inside of our cells, right, what we call endogenous antigens. We want those presented. Oh, my thing goes on the other side. Darn it. Why do I start on the right-hand side? Probably because I'm right-handed. So stuff that we take in, right, and that's only antigen-presenting cells that's actually going to take stuff in. So remember, who's our antigen-presenting cells? We've been talking about them. B cells, macrophages, dendritic cells, right? Those are the only ones that have MHC class 2, right? And they're taking stuff from outside, exogenous antigens. And so remember, all these guys communicate with who? Who does a macrophage communicate with? Who does a B cell and a dendritic cell communicate with? T cells. But which T cells? helper T cells, which are CD4. 2, 4, 6, 8. Okay. So exogenous, right? So stuff from the outside. Endogenous, stuff that gets inside, right, or gets corrupted on the inside. We need to also transfer this information. And we do that by MHC class 1. And so this molecule is found on all nucleated cells, including our antigen-presenting cells. Right? So especially our dendritic cells, they have both. Right? Sometimes we forget because they have an extra one. They have the special MHC class 2. We forget that they also have MHC class 1. So I'm not going to play this one, but you guys can. So T cells play different roles than B cells, of course. They never produce antibodies, right? They must have antigen presented to them by those antigen-presenting cells. Effector T cells, again, they're still having antigen presented to them. It's just by any of our cells, all nucleated cells. And it's bad news for them if that cytotoxic T cell recognizes the antigen they're presenting, because they're going to cause that cell to die, because there's something bad inside. So this one has pictures along with it, so I kind of like it. Right? So endogenous antigen, MHC class 1. So again, we're going to talk to a cytotoxic T cell with CD8. This is going to cause death of this cell. Any of our antigen-presenting cells that take stuff in from the outside, right, the B cell, the macrophage, the dendritic cell, are going to present on MHC class 2, which is going to allow them to communicate with the helper T cell, which is CD4, which is going to do exactly that, right? It's going to help out that cell. So one of the things that we'll do for macrophages, although a macrophage can activate a T cell can also, when it communicates with that macrophage, when they recognize the same antigen, it can help make that macrophage a much greater killer, right? In its essence, basically activate that macrophage further as well, right? So it can be twofold there. Typically, the dendritic cell is just going to activate the the T helper cell. What happens when we activate any of these lymphocytes, a B cell or a T cell? That's the first step. After they've been activated, they're just like, woohoo, yay, I recognize antigen. Woohoo. What's it going to do? Do we want just one? Proliferate, right? That's the number one important thing, that when these cells are activated, when the B cells and T cells are activated, they proliferate, right? You get a whole bunch of them that are all the same. So sometimes it's referred to as clonal expansion. Then they're going to differentiate into doing different things, right? So especially for B cells, now we differentiate into what cells are going to actually secrete antibodies. The plasma cells. Because remember, the rough endoplasmic reticulum gets huge because it's secreting a protein, an antibody. What else do you get besides plasma cells? Memory cells. So the same thing, when a helper T cell is activated, right, you get ones that go around and help, right? What else do you get? Memory cells. When a T cell, a CD8 T cell, right, some of them are going to go hunt out and find cells that are infected or something wrong on the inside and kill them. But we're still also going to produce memory cells. For all your B cells and your T cells, whenever you get activation and proliferation, some of them are going to be memory cells, some of them are going to be effector cells. They're going to go out and do their job, whatever that job is. So say in a lymphoid organ, right, in a lymph node, a dendritic cell picks up something and travels even to the lymph node. If it's I don't like this diagram though. It shows MHC class 1 and 2, right, but as different colors. Do y'all see that? Like this one's brown for MHC class 1 and this is MHC class 2. But the important thing is, again, it can talk to both helper T cells with MHC class 2 or cytotoxic T cells with MHC class 1. That's the only reason why I show this one. That it can do both, right? It can do both. So dendritic cells play a crucial role, right? They reside in our peripheral tissues, our skin, our mucosa. They're going to gather material by phagocytosis and pinocytosis. They can even send their tentacles out in between cells and grab onto stuff. They have toll-like receptors on them, so they can help recognize if it's even a bad guy. If a pathogen's detected, it's going to take in even more, right? It's going to gather as much information as it can. It's like, oh, there's a bacteria here, right? One of my toll-like receptors is recognizing lipopolysaccharide, right? Or peptidoglycan. So he's going to gather all that information. All right, there's going to be additional signaling. Again, he can present either on MHC class 1 or 2. Macrophages and B cells can also present MHC class 2 and produce co-stimulatory molecules. It's a little bit more complicated than what we're going over, right? We're keeping it at the simplistic level, thank goodness, right? There's lots of communication that happens between these cells. We're only focusing on one of the major interactions. So what's the difference between helper T cells and cytotoxic T cells with respect to antigen recognition and the response to the antigen? Well, helper T cells, are they going to affect antigen in any specific way? No, they just help. And who do they help? They help B cells, which B cells are then going to make antibodies. And that's what's going to directly interact with antigen. Cytotoxic T cells, on the other hand, what do? They're going to kill. They're going to kill what? Infected cells, right? Cells that are expressing that antigen. So this killer T cell, right, is a cytotoxic CD8 cell. He has the ability to recognize, right, if your cell is infected with a virus and it's presenting it on its surface in MHC class 1. If he recognizes, he's going to kill him. Right? If it's a bacteria infected, right? Intracellular bacteria. If it's a cancer cell, right? If it's expressing bad proteins on its surface that our cytotoxic T cells recognize, they're going to kill it. I'm going to skip over this one too. So again, that cell is going to go through programmed cell death, right? So it's going to produce proteases that just chew up its protein, cytotoxins, perforin that's going to poke its membranes. It's just going to destroy that cell. Apoptosis means without what? Without inflammation. Although sometimes, depending on what the invader is, inflammation might be elicited. So, recognizing an antigen presented by MHC class 2 from an antigen-presenting cell. We'll activate with cytokines. We may recognize a different epitope. So, for example, a B cell is likely to recognize an epitope on a pathogen's surface because the B cell has antibodies, right, on its surface. It can directly interact with an antigen. So think about this. If you're talking about a bacteria cell, like E. coli 0157, which produces specific sugars on its surface, that strain of bacteria. So if we have antibodies on our B cell that's recognizing those specific sugars, that's something kind of on the outside, right, that epitope. But then when a cell internalizes, like a macrophage or a dendritic cell or even the B cell itself, when it takes in that bacteria, it's going to break it into pieces. and pick up a whole bunch of different pieces and present it on its surface. Some of those pieces might actually be from within inside the cell, right? Instead of just the outside of the cell. In the case of a bacteria, we really want outside stuff, right? Not necessarily interacting to inside stuff. So this can create a problem sometimes too, because T cells, because it gets broken up, a lot of times are interacting with stuff either on the inside or the outside of cells. So in making vaccines, we really want T cell involvement, right? We usually want that class switch to IgG instead of just staying at IgM. In order to do that, right, we need to develop vaccines a little bit differently sometimes. We can make what's called a conjugated vaccine against a T-independent antigen. So this is an antigen, right, that normally we don't need T cell involvement. So the capsule, for instance, of Haemophilus influenzae, we can convert that into a T-dependent antigen by covalently attaching a protein to it. So we take a piece of the capsule, right, which is an outside component, which, you know, B cells could activate on their own and produce antibodies against it. We stick a protein to it so that now we're going to get helper T cell involvement. When a macrophage or a dendritic cell takes in that complex molecule, that conjugated vaccine, it's going to break apart those two pieces, the capsule piece and the protein piece. And this is going to help activate helper T cells, which are then, of course, going to activate that B cell and cause class switching to happen, produce IgG antibodies instead of just IgM. Does that make sense to you guys? A little bit. It's a little trickery that we play, right? I'm trying to think. Pertussis, whooping cough, that we're going to talk about later. The vaccine for that, again, is against the capsule. And at one time, it was causing children to spike really high fevers. So doctors literally stopped giving the vaccine because they got tired of moms calling up panicking that their kids had a fever after this vaccine. Even though they told them they're going to probably have a fever, it's okay, right? So until the developers made changes to that vaccine where you didn't have quite the fever response for that. So they've made changes to that vaccine in the recent past, I guess you could say, to make it so that they didn't have quite that fever response. So they tweak and they change things to make the best possible vaccine. A hapten is a molecule that can bind to a B cell receptor, right, to the antibody on the B cell, but it does not elicit a response unless it binds to a protein that acts like a conjugated vaccine. So this happens with penicillin, like people who ingest penicillin, it actually binds to our own proteins, and then you can almost say it's large enough that it then can cause a response. And so this brings us to what we're going to talk about next is allergy, right? So we're going to get the same response that we do to a foreign thing because that's what penicillin is, right? Our body's saying it's foreign, so therefore it must be bad, right? No, right? Penicillin's not going to hurt you. You don't make peptidoglycan. It's not going to interfere with your ability to do that. There's no reason for our body to attack. But for some people, right, they recognize penicillin and they go against it aggressively like it's a foreign and invading toxin. Right? So the same set of events that we've just talked about, right? You're going to get activation of B cells, you're going to produce antibodies, antibodies are going to bind, and you can get those possible outcomes, which sometimes can be quite damaging to the body if we don't need to do it. Right? That's why it's called an allergic reaction, because it's not necessary for what it is that invaded us. But the same steps that we go through for a pathogen are the same steps that we go through for an allergen. The difference is the substance that got into our body, not going to hurt you. Not going to hurt you. So as I mentioned earlier, right, helper T cells can help activate macrophages. And again, it's that CD4 and MHC class 2, this ability to communicate with each other. And the macrophage, when it's been activated by helper T cells, can increase in size, increase its metabolism, the number of lysosomes that it produces. It'll produce different toxic substances like uric oxide, other toxic compounds. Activated macrophages are the ones that can fuse to form those giant cells. And then when T cells are involved in that whole mass, what do we call that? When you've got giant cells forming and then T cells come in and you get this clustering of cells. Granulomas, right? And remember, this is a common condition that happens with what disease? Tuberculosis. So that's just the steps of the process. So for natural killer cells, right? These are also lymphocytes, right? But do they have an antigen receptor? Do they have an antibody or a T cell receptor? No, because they're not a B cell or a T cell, are they? They're a totally different lymphocyte. And they're different from B cells and T cells in that they do not specifically recognize antigen. They're not antigen specific. But they are quite protective, as their name says, right? They're naturally good killers. But they need help in order to kill. So we already talked about one of their abilities to kill. What help do they need? What has to bind to the surface of the cell first in order for the natural killer cell to kill that cell? Antibodies. Which class of antibodies? Only one of them. GG, right? IgG. We call this special outcome of antibody-antigen interaction antibody-dependent cellular cytotoxicity. Right? You got to have IgG on the surface of the cell. It's the IgG, that class of antibody that's recognizing the pathogen that cell is expressing. Then the natural killer cell comes along, grabs on to what portion of the antibody that's sticking out? The FC portion, right? Because natural killer cells have a receptor for the FC portion of just IgGs. That interaction is going to let that natural killer cell know that that cell must die. So it's going to release, proteases, cytotoxic things, and it's going to kill that cell that those antibodies are bound to. So what was it important? So all our nucleated cells, what marker did we say they have on their surface? MHC class 1. The first one is on everything, right? It's on everything. So when a virus gets into our cells, right, and it's having our cell produce its proteins for its capsid, for its protein coat, right? And what's going to happen is a lot of times our MHC class 1 is going to pick up pieces of those proteins and present them on the surface of the cell. So who we hope is going to come along and kill off that cell? If it's producing MHC class 1, right, it's on its surface, it's a viral protein, who should come by and hopefully recognize that? A cytotoxic T cell, right? An activated cytotoxic T cell. Well, for one reason or another, somehow some viruses have adapted such that they shut down our expression of MHC class 1. So they're in there making their proteins and we're not picking them up and displaying them on our surface. So is the cytotoxic T cell going to be able to help us? No, has no way of recognizing, right, that there is bad stuff inside. It's not being presented. Do we have a backup to that problem? Yes, it's called Mr. Natural Killer Cell. So guess what he does to cells who are not expressing MHC class 1? Because that's not normal, right? Should always be expressing MHC class 1. Right? Whenever you go through security, what do you have to show? Your ID. For our cells, guess what our ID is? Our MHC class 1. You must always be presenting your ID. If you're not, right, you're hiding something. And the natural killer cell is going to take care of you. So those two distinct protective outcomes is it's going to kill anybody who's not expressing MHC class 1. That's not a normal situation. You always have got to show your ID. And then if you're coded in IgG, the natural killer cell could come along and kill you. That's antibody-dependent cellular cytotoxicity. So think about this. When you get an organ from somebody else, are their MHC ones the same as yours? Not unless they're your identical twin. That's the only possibility of someone being genetically identical to you. Because remember, how do you know how to make anything your cells make? It's encoded where? In your DNA. Only an identical twin would have the identical DNA as you do. Who's going to be a closest match, though? A relative, right? Because you're a genetic relative, I should say, right? Because you can be a relative by marriage. That doesn't make you genetically linked, right? Right. A stepsister's not going to help you out, right? That's by marriage. A half-sister, on the other hand, means that you share one same parent, right? So there's a possibility of enough relationship there. That's what they're trying to match, y'all. When they're typing, right, they're doing matches, is these MHCs, because, as you can see, if the body cannot communicate with it, now natural killers aren't going to eliminate it, right, because there's at least MHC there. Question is, does it recognize it? And then you have the whole problem with cytotoxic T cells. So anyone who doesn't get an organ from their identical twin for the rest of their lives are immunosuppressant drugs to stop their immune system from attacking and killing that foreign organ. Because again, it can't communicate with it, it can't recognize it. So the natural killers are descendants from lymphoid stem cells, right? So they are a lymphocyte, but they lack, as we said, antigen specificity. They have no antigen receptors. They can recognize, though, the Fc portion of IgGs, right, and can kill those cells coded in antibodies. They can help with the adaptive immune response in that they are important in the process of antibody-dependent cellular cytotoxicity, right? That's that process of being coded in IgGs, and the natural killer cell recognizes it. And they're also really helpful in viral infection that shut down the MHC class 1 presentation. So this brings us back to the very first website I sent you guys to, right? This cute little guy, a dendritic cell, takes in an antigen, right? And he presents it on his surface. The one thing they're not showing you here is that, what is he probably presenting it in? MHC class what? 2. How'd you figure that out? It's a helper T cell, right? See? It's labeled helper T cell. Which means he has CD4, which talks to only MHC class 2. And also, right, they said antigen presentation. But dendritic cells could present by MHC class 1 as well. It just depends on what type of antigen did it ingest. Where did it come from? Right? So he's become activated. What's the first thing he's going to do? Proliferate. Oh, you guys have got it. I love it. Okay, good. All right. So again, right, just to reiterate, even though they're showing for the dendritic cell MHC class 2 in that picture, they can also do MHC class 1 and talk to cytotoxic CD8 T cells. Okay, so here it says a B cell, right? So what's his first step? Could he activate at this point? He's interacting with a specific antigen, this bacteria, on a specific epitope. Do you think he could go and produce antibodies on his own? Why, yes. If it's a T-independent antigen, what does it have to do if it's a T-independent antigen? Interact with more than... Yeah, it's got to interact with more than one antibody on the surface. Is this interacting with more than one antibody? No, right? So we're probably going to need T-cell help, right? This is probably a T-dependent antigen. So how do we get T-cell help? What does that B-cell have to do? What does he have to do? He needs to get activated. How is he going to get activated, though? The helper T-cell. So how does he communicate, though, with the helper T-cell? He has to actually internalize it, right? And break it down and express it in what molecule? MHC class 2, right? They don't even have it drawn out here, right? They just have T-cell buddy talking to B-cell buddy, right? They kind of left out some really important steps there in the drawing. We'll hold it against them, right? We won't hold it against them. You guys need to know it, obviously, in more detail than they're drawing here, right? Practice, draw it out for yourself so you remember, right, what the interaction would be here. Okay, so once B cell has become activated, what is he going to do? He's going to proliferate. And that's it? Differentiate. Whose cell types are we going to get? Plasma cells and memory cells, right? So plasma cells and memory cells, and I love this cartoon. So what do plasma cells do? They secrete antibodies so that he's spitting them out. It makes me laugh. I think it's cute. But yeah, he secretes the antibodies, right? Which can directly interact with the antigen, right? And they can travel through our blood, right? If it's IgA, it can go where? In our mucus, our tears, our saliva, all that good stuff. All our secretions, breast milk. Right? Help protect the baby's intestinal tract. Okay. So when antigens and antibodies bind, we can get a whole bunch of different things happening, right? What's one thing you would definitely probably want to happen if this is a bacteria and we coat it in antibodies? Is that enough? No. What are we hoping comes along and takes care of this bacteria that's coated in antibodies? A phagocyte, right? So look at that beautiful macrophage. That big eater. He's like, ooh, this one looks delicious. Why does he look delicious? What does he have on his surface? Antibodies. When antibodies bind to an antigen, what do we call this protective outcome when it's basically tagged for destruction by this phagocytic cell? Opsonization, right? So he's been opsonized. And the important thing here, again, is what portion of that antibody is that phagocytic cell going to grab and onto and recognize? The FC region, right? This region sticking out, I know it's hard, this is a small molecule, right? But he literally is going to grab onto that. And then we're going to gobble up, hopefully, and eliminate all those bad guys, right? All those bacteria. You know, if this is in our mucus, it can maybe help trap them so that the mucus can keep them in the mucus and push them away from the body. Also remember, when an antibody binds, can that bacteria bind to our cells? No, we stop them from adhering. That's one of the protective outcomes of antibodies, right? We can clump them up too, right? We can cross-link, we can agglutinate them. We can clump them up, keep them all in one area. We can opsonize them. The bad guys are going to come in and grab them. We can stop them from binding to us. And if this is IgG, who else might destroy this? Natural killer cell would seem like, right? But the problem here, and one of the things you may not have noticed earlier with natural killer cells, is natural killer cells are attached to our cells. They attack our cells. So that protective outcome of natural killer cells doesn't work against bacterial cells, fungal cells, right? Algal cells, although none of them are pathogenic, but, you know, some other foreign cell. That is a protection against our own cells being infected or defective. It's a cellular response. So, we clear up all the bad guys. Do they come back again? Potentially they will, right? You could see that bad guy again. So this was the very first time, say, we saw that pathogen. So we produced memory cells, right? This B cell that was activated may have been what we call a naive cell. What does that mean? What does naive mean? It's just out of the thymus. It's not activated yet. All of those are correct. There's one more thing I'm looking for. What does naive mean? Like, think about that word. No experience, unaware. Unaware of what? What's important to these guys? What are they unaware of as of yet? The bad guys, right? The antigen, right? If they're naive, they've never seen their specific antigen yet. Once they've been activated, obviously they've seen it and they know it. And we produce memory cells. So that way, the second time we see it, are we activating naive cells? We could. But the good news is our response is usually faster because we're not trying to find a naive that recognizes. Instead, we already have who that already has seen the bad guy? The memory cells, right? And that's why, again, our secondary response is so much faster and aggressive than the first response. This is important, y'all, because guess what? The very first time you see an allergen, you make what against it? You could make IgE, yeah. If it's an allergen, typically the problem is one, that we're making that particular class of antibodies and what it binds to. The fact that you made antibodies, what else did you make? Memory cells, right? So you become what's called sensitized. Your body, you found, unfortunately, naive B cells and T cells because you made a class switch to IgG if you're dealing with IgE, excuse me. Erica absolutely hates it. I don't even want to say it. I hate it so much. I get too much IgE. It's too many things. Right? So the problem is you've got these memory cells too. So each time you're exposed, guess what happens to the response, y'all? It's faster and greater. Right? Because your body's like, oh, it's back again. Right? We gotta aggressively get rid of it again. Problem is not a bad guy. It's a pollen granule. It's not going to do anything. Body doesn't know that. All right, so that's actually, I'm going to